Oh, wow. Good morning or afternoon, depending on where you are. Good to see you. Piling in. Wow, welcome to Club Ev Med. While we're waiting for everyone in the waiting room to join us, wish you a Happy New Year on behalf of the International Society for Evolution Medicine and Public Health. It's good to see you all for the first Club EvMed conversation of 2024. The aim of these Club EvMed conversations is to connect the community of people who are working on questions that intersect between evolutionary biology, public health, and medicine, and to share exciting new research. They are conversations, so I encourage you to ask questions during the talk. Our speakers today are comfortable with that. Alternatively, type your questions in the conversation in the chat box, chat with each other using the chat box, converse how you'd like to converse. We like to get a range of perspectives and questions. If you haven't renewed your membership to ICEMF, now is the time to do it. Club EvMed, the event today, is organized by 8 evolutionary medicine centers and programs. And from Latin America, Brazil, from Europe, including Germany, the US, including ASU, Vanderbilt, Duke, UNC, a number of centers. So we've got a broad audience, and we're welcoming you to please participate as much as possible. Today's talk is a part of our series called Translated Winds in Evolutionary Biology. That is exciting things news you can use from evolutionary medicine. And our speakers today are Dr. Tony Capra and Dr. Keila Velazquez-Arcelay, both from San Francisco. Tony is from the Department of Epidemiology and Biostatistics at UCSF. And Keila is based in San Francisco and is a new PhD from Vanderbilt. You can join the list for Club EvMed by clicking on the link in the chat. Right now, let's turn it over to Tony and Keila, who will talk about how archaic introgression shaped human circadian traits. Something that is relevant to everyone here. Take it away.
Hi, everyone. It's great to be here. Thanks so much for the invitation to come and speak. I just want to make sure everyone can see and hear all right. Great. So before I get started, I want to start just by saying thank you to the organizers, but also to everyone that helped out with this work. Since this is a conversation, I don't want to be rushed at the end trying to fit this in. I want to do this at the start. So you'll get to hear from Keila, but this project was integrated many different skills and techniques and perspectives from folks in my lab over the years, including David Rinker, Laura Colbrand, Colin Brand, and Yvonne MacArthur. as you'll see, it also involved a sort of essential contribution from Doug McMahon and his postdoc, Justin Seaman, who's a circadian biologist at Vanderbilt. And that proved really, really essential to us making sense of some of the things that we were seeing. So thanks to all of you for being here, and thanks to all of them. So I'd like to start by one other sort of side point that maybe at the end, if there's time, we can also talk about. And that's some interesting lessons that Keila and I have learned about science communication as a result of this paper. So we published this manuscript in GBE sort of in mid-December, and it sort of unexpectedly generated a ton of press interest. And some of you may have seen headlines like, morning person, you might have a Neanderthal gene to thank, or Neanderthals were early risers. And so if you're here hoping to be told that, I'm sorry, I'm going to disappoint you that pretty much none of these claims are what we were saying or what we said in our article. And it just has proven really challenging to sort of communicate some very complex evolutionary biology in a way that doesn't get distorted in the popular press. So like I said, at the end, if there's time, I'd love to hear people's thoughts and experiences about that. But I'll focus on what we actually did see. So I think probably everyone here is familiar with the revolution that the ability to sequence ancient DNA has really produced in our understanding of recent human history and migration. We now know, of course, that there were all sorts of interbreeding events between different groups in the recent past. We even know about groups that, like the Denisovans, that we really didn't even have a clear idea existed before. We also know that the human populations were very, very dynamic and mobile, moving, often on the scales of just a few hundred or thousands of years over broad distances. And so, the ability to sort of see the genetic relatedness back in time has been really, really powerful and influential in our recent understanding of human evolution. But, my lab is really interested in a related question, but one that I feel like ancient DNA has not yet fully achieved its promise in, and that's, what were these individuals living in the past like? And you might appreciate that obtaining ancient phenotypes beyond the DNA sequence is very challenging. Here's a picture of the Denisova cave in Siberia, where many of these Neanderthal Denisovan samples have come from. It's not a very hospitable place, and so you can manage to extract DNA from these materials, but you can't do RNA-seq or any of the other functional genomics types of methods that we are commonly using on modern samples. And of course, you're not going to see anything about other more organism level traits than just what you can infer from the bones. So this is a challenge, and we'll talk about some methods that we've been using to try to get around it. We're also really interested in the question of how these other groups influenced us, modern humans. And we now know that as anatomically modern humans migrated out of Africa, they interbred with the Neanderthals, and then further on with Denisovans. And this was quite interesting because these groups had been living over broad ranges in the Mideast, in Europe, and Central Asia, and Southeast Asia for really often hundreds of thousands of years before anatomically modern humans ever got there. And so there's the potential that they helped modern humans, that this introgression or interbreeding helped modern humans adapt to these unique environments. And indeed, there have been several examples where this has been shown to likely have been true. And one of the most striking, and I think unexpected ones, was that altitude adaptation in Tibetans was likely contributed to by DNA of Denisovan origin. And here's a haplotype tree from that paper back in 2014 that shows, each of these is a represents a DNA sequence haplotype. This is around the EPAS1 gene. This will be close to many of your hearts and minds. And what we see down here in the corner is that the Tibetan haplotypes that are adapted to their high altitude lifestyles are actually most similar here and look to have been derived from a Denisovan haplotype. So just really, really a fascinating example of where some variation in this other group that had separated from the anatomically modern humans hundreds of thousands of years in the past proved to have an adaptive benefit. And of course, there have been many more candidates for this kind of adaptive introgression, in particular in terms of hair and skin traits, some immune system traits, and some metabolic traits. And it was sort of with this background that Keila started thinking, what about latitude? And I'll let her pick it up from here.
Yeah, and so we were motivated by these traits that were influenced by Neanderthal introgression into humans. And so we decided to explore the effect of these archaic alleles on the circadian biology of humans living at higher latitudes, because as you know, different parts of the planet have different patterns of season and yearly light exposure depending on latitudes. And we wanted to see the effect of that on circadian biology. Next. So circadian rhythms are mainly composed of a negative feedback loop, where in humans, the clock and BMAL protein interact to activate the expression of the system, while the period and cryptochrome proteins they repress the expression of these genes of this system, and this expression oscillates throughout the day. And this clock system, it controls the timing of many biological functions, including metabolism. And it is known that a misalignment between the internal clock and the external patterns of behavior or environment, they can cause various health risks, including cancer and obesity. So as we mentioned before, different latitudes of the globe receive different patterns of light and dark throughout the year. And although the effects of latitudes on circadian biology is not well understood in humans, we know from other animal systems that this can have an effect on allele frequency and behavior in these individuals. For example, in Drosophila, is a model organism who study circadian biology, and they happen to have had a similar migration history that humans had, migrating from Africa to other parts of the world, and a frequency differences in alleles in the TIM gene have been observed, and this allele, it is associated with a more independent free running clock at higher latitudes, and improved diapause. And then these alleles have also been observed to affect the length of the circadian clock. It can make them shorter or longer. So it can also affect the behavior of the Drosophila. The variation in timing of circadian expression contributes to different chronotypes, which is the time of day when an individual is more alert. And the UK Biobank data contains information about the chronotype of some of these participants. So these data were collected to classify individuals in 4 different chronotype categories, ranging from definitely a morning person to definitely an evening person. Next. So based on this data, studies have been able to identify the heritability of genetics on chronotype. Different studies have done this on twins, and the heritability has been estimated from 12 to 38%. Another study using this UK Biobank data identified different human traits that influence that are influenced by Neanderthal alleles, and some of those traits that were significantly influenced include a non-coding site near the gene ASB1, and this variant has been identified as affecting chronotype in humans. Also, the study identified a latitudinal line in this allele across Eurasia. So based on these observations, we wanted to study the difference between human and circadian biology as the first question in our study. And then we also wanted to study the contribution of Neanderthal alleles in human circadian biology. First, to be able to explore these questions, we created a set of genes that are involved in circadian biology. And this set includes the core clock genes and also other genes that interact with the clock circuitry. Then we also identified flanking non-coding regions from those genes that interact with circadian genes. But circadian biology is complex, so this list is likely incomplete. To answer our first question about the difference between Neanderthal and human biology, we studied genetic variation in our regions or circadian genomes regions that are specific to each lineage, specific to the human lineage or specific to the Neanderthal lineage. We then evaluated this lineage-specific variation in circadian genes and also in the promoter and regulatory elements nearby. And we found that the proportion of introgressed variants found in this region is larger than expected. So this suggests that there was something happening here in the interaction between the environment and circadian biology of these individuals evolving in different environments at different latitude. But then we also wanted to explore what the phenotypic effects of these variants is on humans.
So I'll pick it up here for a couple slides. And as I was telling you before, there's things it's hard to infer from what remains, and certainly something about the circadian clock or even a behavior like chronotype. It's not clear how we can get that just from the fossil record. So a big theme of the work in my lab is thinking about what we can infer about phenotypes from ancient genomes. And so I want to introduce an approach that we've been taking in many different projects and that we've applied here as I think a really nice case study. And this approach has been enabled by advances in machine learning over the past 5 years that are really enabling the increasingly accurate prediction of molecular phenotypes from DNA sequence alone. And now many of you might be familiar with polygenic risk scores, where genetic variants along the genome are used in a linear model to try to predict the setting of some phenotype in a particular individual based on their genome. And often this is used in sort of the estimation of risk for common complex diseases. But these approaches really do not generalize very well, even across human populations. So we're reluctant to apply those kinds of approaches. And the approach that we've focused on has been to try to predict informative molecular phenotypes that are likely based on much more conserved and easier to predict aspects of biology. So for example, many of you are likely familiar with AlphaFold 2 and Rosetta Fold and ESM Fold and all these new variants of language models that are being used to predict very accurate protein 3D structures just from protein sequence alone. But there also are increasingly accurate methods for inferring RNA or gene expression levels just from looking at DNA sequence. And so many of these approaches have been developed in the context of transcriptome-wide association studies like PredictScan that essentially learns a linear combination of weights for genetic variants near a gene. Other more complex deep neural network models like Informer and Splice AI that attempt to just look at the whole DNA sequence and infer the expression level or patterns of splicing. And there are also methods that attempt to infer something about the cellular or chromatin environment, like Akita and Orca and DeepSea, that attempt to learn patterns of 3D contact, like essentially the 3D structure of DNA in the genome. And this is really exciting to us because it gives us an opportunity to infer these ancient traits from genetic sequence alone. And so the basic idea is this. We have an algorithm that we train using data from modern human populations. For example, let's say these little squiggles represent mRNA levels. And then we could use any one of a range of machine learning techniques just based on whatever we think is appropriate for the problem. And then we can take as input a DNA sequence from a genome of an ancient or archaic individual and see what the model says would be the output for that DNA sequence. And then we can look for differences by taking, for example, in this case, the three high coverage Neanderthal genomes and the one high coverage Denisovan genome, putting them into the model and predicting whatever molecular phenotype that model is trained on. And then we can take a diverse array of modern humans, and usually we do this in humans that do not have introgression because we want to sort of see that independent sort of human state, get a distribution of modern humans and look for cases like this, where the archaic hominins fall outside the range of what we see in modern humans, okay? And so we've applied this general strategy to identify divergence in gene expression and splicing and genome folding, all using different techniques. And today we're going to focus on some results from these methods for inferring divergence in gene regulation and splicing. But I do encourage you to check out other papers we have that have sort of investigated these strategies more broadly without a specific trait focus. And so one thing I hope you'll take with you is that these sequence-based machine learning algorithms are providing a really exciting way to generate hypotheses about molecular divergences between modern populations and ancient or archaic individuals. And so we asked essentially, are any circadian genes differentially regulated between humans and archaic hominins based on these DNA sequence models? And at this point, I'll turn it back over to Keila to show you what she found.
Yeah. Thanks. Less than 7 free long.
Oh yeah, everyone please turn off your mics. Thanks.
Okay, using the methods mentioned previously, we were able to identify circadian genes that have evidence of being divergently regulated between humans and the Neanderthals, and this includes 4 circadian genes. And as you can see here, the predictions of regulation in Neanderthals falls outside the human predictions. And in summary, using these methods, 18% of the genes in our circadian set are either divergently regulated or contain alternative splicing altering variants in archaic hominins. So with this result, the answer to our first question of did circadian biology differ between archaic hominins and humans is likely yes. And then following this observation, we wanted to see if the introgression of archaic alleles contributed to human circadian biology. So to be able to understand the contribution of Neanderthals, we created a set of circadian introgressed variants from a general set of introgressed variants that were identified by the S' method. And from that set, we only considered introgressed variants that were supported by the S' method and at least one other methods for robustness in our study. Using that set, we looked for the effect of introgressed variants on the regulation of circadian genes, and we considered over 4 million EQTLs from the GTEx project. And for each EQTL, we asked, is this EQTL introgressed or not? And if it is it associated with circadian gene or not? And following that analysis, we found nearly 4,000 variants falling into this category, and an enrichment analysis results in a 1.45-fold enrichment of introgressed variants in circadian genes that are EQTLs, or expression quantitative trait loci, showing that introgressed variants are enriched in this area. And these results also show that this enrichment is consistent across the different tissues that are studied in the GTEx catalog. So next, we wanted to understand the influence on introgression on phenotype, specifically on chronotype. So to do this, we considered GWAS results from the UK biobank that we saw before, and we applied stratified LD score regression to the summary statistic from this data, and introgressed alleles that match Neanderthals. And well, stratified LD score regressions, the estimated proportion of heritability of a trait contributed by a partition of variants of the genome. And following this study, for various phenotypes, and one of those phenotypes includes chronotype, specifically the morning person trait from the UK Biobank. So using that method, we were able to estimate the contribution of introgressed variants to heritability. And also, additional to that, we were able to see a direction of effect in this enrichment, and the enrichment goes in the direction of morning person enrichment for Neanderthal introgressed alleles. So we wanted to explore further this observation, and we studied a cumulative fraction of the effect of these variants on the effect of morningness specifically in these individuals in the UK Biobank, and we found that these circadian variants consistently increase morningness in these individuals, but the effect is even stronger when you look only at alleles in or variants in our set of circadian genes instead of the general variants in the genome.
And so now this, to me was really surprising and striking to see such a directional bias in the associations of the introgressed variants with this phenotype. And normally we don't see that for other traits. Usually it's sort of a little more balanced. And honestly, Keila and I sort of scratched our heads about this and thought, well, we don't really know. We can't come up with any good reason why Neanderthal variants would more likely increase the proportion or more likely increase your propensity for morningness. Fortunately, we had expert in circadian biology very nearby, and could just ask him. And as soon as we sort of showed these data to Doug, he's like, "Oh yeah, that makes total sense." And we said, "Okay, well, why?" And he said, "Because in humans, early risers, so people that are morning people, have shorter circadian clock periods. So in essence, how fast that feedback loop Keila showed you earlier runs a bit faster in morning people. And connecting, and I think that the key step was Doug was able to sort of synthesize these results with Drosophila as well. And in Drosophila, shortened circadian periods are required for synchronization to the variation in light levels at higher latitudes. Okay, so shortened periods are ending up having this role in synchronization to the latitudinal cline. And it appears that in humans, shorter periods are associated with being a morning person. And so also in Drosophila, it appears there's been selection for shorter periods that's resulted in latitudinal clines of decreasing period with increasing latitude. So again, this points to shorter periods being beneficial at higher latitudes, and shorter periods increases the propensity for being a morning person. And so thus, this directional effect of introgressed circadian variants on chronotype towards being more of a morning person may not actually have anything to do with waking up early, but may actually be about the shortening of the clock that appears to be beneficial in having a more a flexible clock, if you will, that's easier to entrain to those varying light levels. And so, this is consistent with a latitudinally adaptive pattern that's been seen in Drosophila. But of course, there are many logical steps here that we have not yet worked out that they really do connect. And honestly, that underlying this connection in humans, I think it's not totally understood why that is, that the shorter clock necessarily leads to morningness. But I am far from an expert on the circadian biology, so forgive me if that's wrong. So to summarize, we've showed that there was a lot of genetic difference in parts of the genome that are relevant to the workings of the circadian clock and downstream processes. And moreover, that some of that variation that was introgressed from Neanderthals into modern humans contributes to variation in human chronotype. And it does so in a direction that is consistent with what we've seen in adaptation to higher latitudes in other species. And so again, this is we haven't proven that it was adaptive, but it is at least consistent with that argument. And so with that, I'd like to mention several limitations of what we've done. So first of all, our analyses were mainly focused on European ancestry cohorts. And that's because that's where the large GWASs and functional data on chronotype and circadian biology come from. And so we are working to identify other cohorts and other biobanks where we can see if these patterns hold in other populations. As I was suggesting before, this is consistent with introgression being adaptive, but this is always very challenging to argue. If you look at, so we applied several different methods for identifying adaptively introgressed loci and found that actually 47 different introgressed loci near circadian genes have evidence from at least one of these methods of being adaptive. But these methods didn't agree on any of them. We found several that were good candidates that had a couple of them that agree. But again, this is just a very, very challenging signature to detect from just genetic variation alone. And especially if the selection is on a very polygenic complex trait like this, that there are many different genes that have different effects on the clock and its downstream effects. Another, I think, obvious limitation is that we can't prove that the lineage-specific variants produced functional differences, at least in the Neanderthal lineage, because we don't have Neanderthals that we can ask about their chronotypes. But as I'll tell you in a minute, we're starting to work on some model systems where we might actually be able to get that question. And then I think finally, and perhaps most importantly, is that circadian biology is extremely complex and is linked, as Keila showed you, with many, many systems in our body. So these observations could be totally right, but it could be actually that the selection wasn't acting on the clock itself, but some other aspect that is associated and regulated along with or by the clock. I mean, for example, something like temperature. In general, we just really need more study on how these processes, both in humans and in model systems, interact so that we can sort of make some of those links between the molecular biology, the running of the clock, and then the behavior more clear. And so, as I said, I'm really excited about sort of building on this hypothesis and seeing what we can test in model systems. And so we're working with Doug to try to adapt this endogenous circadian reporter system that was recently published so that we can essentially Neanderthalize the clock in a cell, and by putting the Neanderthal variants that we say are having specific effects, and then see if indeed they do change attributes of the clock, such as its period or its amplitude, in ways that are consistent with the model that we're proposing. And so with that, I just want to again say thank you to the organizers for giving us this opportunity to come and speak. Huge thanks to Doug for his perspective and guidance. And Doug, Laura, Colin, David, Laura, Colin, Yvonne, and Keila. This was such a fun project. And if you think this was cool and you're looking for a postdoc, we are hiring. If you know anyone who is, we are hiring, let them know. And I'd love to work with you. And so it seems like we've got a good amount of time for questions. So please, I'll let the moderators organize, but I'm happy to chat for as long as we can.
Thank you. That was fascinating. The circadian biology is really interesting. So, who has a question? Please feel free to, I don't see hands up. One question that I had was, what is the imputed regulatory score? What was it called? Imputed regulatory function?
Yeah.
What that meant.
Oh, yeah. Thank you for asking that. You know, in the interest of time, we didn't go into detail, but essentially, that is a prediction based on DNA sequence of the genetically regulated component of expression in a given cell type based on that sequence. And so that scale you can think of as being sort of normalized to the distribution of expression of that gene in a human cohort. And so if we see that the archaic hominin is far off of that distribution, in a positive direction, it means that the genetic variants that they carry are pushing them towards higher expression. If it's in a negative direction, it means it's pushing them in a direction of lower expression. And that's based on a model that was trained in sort of modern humans to go from DNA genetic variants to expression levels. Does that help?
Yes.
Cool. Thanks.
Gunter, please ask your
Thank you.
question.
Thank you for this interesting talk. I would like to understand a little better which aspects of your study was misrepresented in the media reports, because I hear you saying that Neanderthal genes make you a morning person, and that doesn't seem to be different from what's in the media.
Well, yeah. So, you know, Keila mentioned the heritability of this trait, and so being a morning person is an incredibly complex trait with likely hundreds of loci, maybe even more, where genetic variation influences your particular, your particular setting of this trait. And so it's really sort of wrong to say that, one variant makes you a morning person or another variant doesn't make you a morning person. And it also depends on which particular Neanderthal variants one might have inherited. So, there were 3 that, and in fact, it's one of them is that first identified one that actually increased propensity for being an evening person. So it's not that every Neanderthal variant makes you more likely to be a morning person. But I guess the core of it is just that, the overall contribution of these variants to this trait in modern populations, is relatively small compared to all the other factors that influence it. Does that make sense?
Okay, yeah, but of course it's a subtle message that maybe journalists have no business to convey.
Well, I would love to hear people's thoughts about that, because the experience that we had, Keila and I talked to reporters, and we explained that to the reporters, and the ones that really cared, I think, got that and described it accurately, somewhere in the article. But then the editor writes the headline for the article and wants to get you to click on it. So often it gets a little bit sensationalized. And we definitely are sort of struggling with that a little bit.
Okay, thank you.
Thank you. June, would you like to ask yours?
Yeah, thank you, Dr. Bell, and thank you, Dr. Capra, for the amazing presentation. I actually have a quick question about the performance, or kind of the way how we gauge the performance of the model that we're using. So particularly, I'm wondering about 2 aspects, right? One is whether the different allele frequencies of the variants of interest, or the haplotype frequencies of the region that we're interested in, will affect the performance of the model. I guess that's the first aspect. And the second aspect is, are there any specific genetic regions that are, whose effects are better predicted by the model, say either intronic or regulatory regions versus exonic regions. And I know sometimes for certain models that are sequence-based tend to predict certain motifs or certain specific sequence that it basically kind of overweights that particular sequence and then kind of predicts something solely based on that combination. I was wondering if we have tested for that, and do we kind of foresee problems like that interfere with the prediction of the Neanderthal genomes?
Yeah, that's an excellent question. There are a number of sort of moving parts in there that I'll try to separate out. So the first was about the performance, I believe, of the sort of sequence-based predictive models that we use to try to identify divergence, whether it's in expression or splicing. And yes, it does vary from gene to gene, for sure, but we have a decent understanding of that, at least in human populations, of where predictions can be made accurately and where they can't be made accurately. And we have a little bit of understanding of that about the introgressed variants, because about the Neanderthal variants, because these models are nice in that we can apply them to the genomes of people that do have Neanderthal ancestry at a locus and people that don't have Neanderthal ancestry at a locus, and compare their accuracy between those two scenarios. And when we've done that, we've found that actually the decrease in performance is pretty small for most of these approaches when applied to people that are Neanderthal-like versus people that are human-like. On the order of 5 to 15%, depending on the model. But based on that and respecting just the sort of challenges of this, we generally don't place too much stock in the sort of the exact magnitude of a change, but are really just looking for cases where there's a really extreme pattern like the ones that Keila showed you, where the archaic hominins are totally outside the range that we see in modern humans, suggesting that there's been something substantial that diverged. And then the hope is you can then go validate hypotheses about specific variants that you think are causing it more in a more targeted way. And in general, we don't fully understand, I think, all the biases and potential of these models. So that's like why we didn't do any sort of enrichment analysis on those divergent regulation predictions. Does that answer your question?
Yes. Yeah. Thanks.
Cool.
Thank you. Kendra, would you like to ask your question? Or would you like me to read it?
Hello?
Yep, we can hear you.
Okay, cool. Let me go read my question again. So I was wondering, was there any genetic variation associated with the circadian rhythms that differed among the archaics? Because you're including both Neanderthals and Denisovans.
Yeah.
As well as oh, sorry.
Oh, no, go ahead.
Oh, and then the other part was, were there any potentially introgressed alleles that were shared, or sorry, were the potentially introgressed alleles shared among all archaics, or like was there any that were like specifically to one archaic or two or whatever?
Yeah, that's an awesome question. Let me see if I can run back to a slide here to illustrate this. And Keila, also, I'm feel free to jump in if I'm monopolizing this. One really interesting thing we discovered was that older archaic variants are much more likely to be affecting traits and to actually be enriched for effects on some traits. And so in essence, we're showing here basically the contribution of all of the introgressed variants to a representative set of about 40 traits versus the contribution from a set that match, and we're getting a little bit deep into the weeds here, but in this case, the Altai Neanderthal, which is a Neanderthal that was quite diverged from the one that introgressed into modern populations. And so in essence, you can think of this axis being those old variants that were shared among likely all Neanderthals and even potentially Denisovans. And then these are a mix of the old and the new that might have been specific to that lineage. And so this is a theme we've been seeing in a lot of different projects, is that those older ones that have been seen by selection longer are more likely to affect phenotype and more likely potentially to be adaptive in modern populations. So That's the first thing that comes to mind. The second was, as you see here in the predictions for the differential regulation, there definitely were cases where the two Neanderthals were more extreme, for example, than the Denisovan, and vice versa. I don't remember the sort of total sharing. I think we've got a Venn diagram somewhere of that. I don't know, Keila, do you remember the exact details of how many were Neanderthal or Denisovan specific?
I don't think we included that one. We did have a figure for that, but we did not include that in the final
Okay.
report.
But no, it's a great question because these individuals were quite different and quite diverged. So I think there's more to learn from teasing apart their differences too. Thanks.
Thank you. Taurus, would you like to ask your question?
Sure thing. First of all, great talk. I've always thought this is a really creative way to get at and infer characteristics of archaic hominids. I'm wondering what evidence or what data sets would you need to actually say something about selection on these phenotypes in the archaic hominids versus just that they are different than modern humans?
Yeah, that's the sort of $1 million question, I think. I would want a combination of lots of pieces of evidence pointing in a consistent direction. And so we have a few of those now. We have what we're seeing in terms of this direction of effect in modern humans making sense with what we think we understand from adaptation in the model organisms and also then in some lab experiments. But I think I'd want that connection between the molecular effect on the clock and then the behavioral effect to be a little bit tighter than we currently understand it. I also would hope to see that there would be some sequence signatures around the regions that we're looking at, the introgressed regions that we're looking at that are consistent with adaptation. But again, like from my perspective on that, it's a really, really hard thing to detect given the nature of that process. And if you compare the methods that are out there for doing it, they just disagree a lot. And so I think for me, the biggest challenge, though, even if all that agreed, would be saying that it was definitely on some specific aspect of all the processes that are related to the clock. So, because we could show this sort of like molecular effect of the introgressed variants on the clock, which we're trying to do, but that still doesn't tell us exactly which potential downstream effect was likely selected. Does that make sense? If you've got an idea about how to do it, I'd love to
Yeah,
hear.
I don't have any ideas either, but it's great question to think about, and that's ultimately I agree it is the $1 million question. Thanks for your thoughts on it.
Thank you. Yeah.
Thank you. Sean Reed? Sion Reed? I may be pronouncing this person's name wrong. I apologize. It's in the there you are. Would you like to ask your question and tell us how to pronounce your name?
Kian.
Ian. Okay, no wonder you didn't respond.
Yeah, I was just wondering, do you think, we know that the immune system is tightly regulated by this circadian rhythm? If they measure different cytokines in serum at different times of day, or they've shown if you get vaccinated at different times of day, it can affect the response. So I was just wondering if you think that these variants could potentially be having an effect on the immune system and how it's regulated.
Yeah, absolutely. I think that's a great example of what I was talking about before with it could be that we're actually capturing some signature of selection on something else that happens to have used a circadian process to enable it. And we've got some ideas about trying to it turns out these variants that we found with effects on chronotype in modern humans are actually fairly pleiotropic. And Keila did a nice analysis where she looked at other associations. And indeed, there was a lot of blood and immune related traits that are associated. So that potential is definitely there for many of those introgressed variants. Like we do see that there are effects on those traits in modern humans. What we're struggling with now is thinking about the right way to detect whether that's more or less of those effects than we expect. Because there's a really strong bias, for example, in the UK Biobank, in terms of the phenotypes that are covered. There's a lot of molecular bud-based phenotypes, so of course we see a lot of them. I mean, I think it's cool that we see a lot of them, that there are associations at all, but I don't read too much into the fact that we see, I forget what the total number is, but many of those immune phenotypes. So I think it really could be. We don't know how to figure it out yet.
It could be some sort of disease maybe that that evolutionary selected for some of these variants could be a cool angle for sure. How you'd analyze that downstream, I have no idea, but yeah.
Yeah, thank you.
Bernie, you had a question. Would you like to pose it?
Well, it's mostly been addressed, and it had to do with selection, as Cian just talked about, that selection in the context of infectious disease would be expected to be sort of a prime candidate for introgression, and I think that's been shown in Neanderthals. And I did notice that autoimmune disorders on a slide that you just showed appeared to be one of the traits that's associated with the introgressed alleles. And when you see autoimmune disorders, I think what you're really seeing is secondary effects of infectious disease selections. I'm just not sure if there was a way that you could look at your introgressed alleles in the context of, well, are they associated with infectious disease risk or not?
Yeah, no, that's an excellent point. I'm glad you bring this up, because the sort of same methods that we use to identify this chronotype enrichment really brought up a lot of autoimmune and other what I think are likely traits related just to the activity of the immune system in response to infection. And there have been a couple of nice studies from other groups where they've, for examples, molecularly worked out, what are the effects of the introgressed variants and why might have those been adaptive in certain environments or certain exposures, in particular to RNA viruses. The challenge I face, and I think, so maybe what you're saying is the null model that we should have is that this is all just due to infection, and can we show that there's an effect beyond what we would expect on that on the circadian clock? And the challenge, I think, is just coming up with the right null model and the way to represent that. So
I wouldn't call it a null. I would just call it a guide to what experiments one might want to do, what data one might want to collect specifically.
No, and I think you're right. Like, as we're considering these downstream effects of changes on the clock itself, whether it's the amplitude or the frequency, we need to keep in mind what effect that would have on the immune processes that are regulated by that clock, as well as the effects on sleep-wake and metabolism. Yeah, great point. Thank you.
Thank you. I think we have time for one last question. Terrence Taylor, would you like to pose your question? Are you there? Well, I'll read it for you then. It says, thinking of the wider implications of your research path, what do you think would be the next step for understanding the evolution of other products of the interaction between ancient hominids and modern humans? Oh, wow, this is a tough question. It also asks you to take into account methodological challenges. And it also focuses on the immune system.
Yeah, well,
okay.
I'll do my best, and it will be inadequate to rise to the challenge of this. But the approach that we are taking is to try to use the methods that we know and like, which are these machine learning methods from computer science that let you make more and more accurate predictions from DNA sequence, and to use those as really hypothesis generation tools that say, we think this variant or this set of variants that have a consistent effect that we can test in large scale genetic data sets, as we did here, might explain some aspect of this interaction or some functional outcome of this interaction. And then the next step is trying to validate those predictions to confirm that they do indeed they are indeed correct, and that they do indeed sort of mechanistically connect to that higher level effect on the organism. So, a genome-wide association study like the ones that we've used here links a Neanderthal variant to an organism level trait, like, do you wake up early in the morning? Do you like to wake up early in the morning? And what I think we need to do is better fill in those gaps up the mechanistic chain from, the genome to the genes that are affected, to the cell types that are affected, to the tissues that are affected, up to the organism. Because then, like, that understanding, I think, will help us see if our hypothesis is consistent at each of those levels. And that's the approach we're taking. I think there are probably others, but it's I think it's going to take these different perspectives to really get at this this interaction, because unfortunately the materials that we have left from them just are not sufficient for a lot of the questions and phenotypes we care about.
Thank you very much, and thank you so much for your excellent presentations, both of you.
Thank you.
That we all want to go from the gene to the organism to the population. So keep up the good fight. Thank you all very much. And thank you, Tony and Keila, for a great presentation and questions.
Thanks, everyone. This was really fun. Please feel free to follow up if you have any other questions.
Okay,
Thank you.
Thank you.